Hello boys, welcome back to another session of video tutorials, and this is meant for class 12th biology students, and this is for 12th October 2020. We are into the revision mode, and this session we are going to revise a very important chapter having maximum marks in your CBSE board exam for your batch, that is molecular basis of inheritance. If you may recollect from your memory, when I was dealing with this chapter on videos, I was telling you that this chapter is very important chapter for all the biology boys. The reason is that it is not only helping you to get good marks in your CBSE board exam as well as in another national level exams like NEET, it gives you career opportunities. That is, you can become in future a molecular biologist, and who knows, you can work with Nobel laureates also. So what is required is personal interest. You need to take lot of personal interest, study the material, understand the concept and practice the concepts. Then only you can aim big. So it's not about just casual reading. I want you to focus and read this material. So with that note, we move ahead. DNA. So the molecular basis of inheritance. What are the two molecules that help in our inheritance? Transfer of characters from parents to offsprings. So two molecules we are referring. One is DNA, another is RNA. Expansion of DNA, D for deoxyribo, N for nucleic, A for acid. Deoxyribonucleic acid. If someone asks you what is your secret, all of us know, biologists say our own DNA is our secret because that makes you our phenotype as well as genotype. What about RNA? Ribo, R for ribo, N for nucleic, A for acid. RNA is ribonucleic acid. I know the present generation, all of you wanted to be muscle builders, and muscle building is possible only when there is a lot of proteins. And who is responsible for protein synthesis? It's RNA. So one is the secrets of our genotype and phenotype. Another is for our appearance, for our strength, for our stamina. RNA is important. So these are where they are present. These are present inside the nucleus of our cell. So they are called nucleic acids, and they are acidic in nature. So having present in the inside the nucleus and having acidic in nature, they are called nucleic acids. So they are found in all living organisms. DNA acts as I said just now, genetic material in most of the organisms. RNA also acts as genetic material in some organisms like COVID-19 viruses particularly, and act as a messenger. Its function as adapter, structural, and in some cases as catalytic molecule, catalysm, breaking of substances, catalytic molecule. The DNA, it's a long polymer of deoxyribonucleotides. So what is DNA? Polymer. It's a polymer, means made up of many monomers. What is the monomer unit here? Ribonucleotide. Deoxyribonucleotide is a monomer. So many deoxyribonucleotides joined together makes a polymer, and that is DNA. So a pair of nucleotide is also known as base pairs. So length of DNA is usually defined as number of nucleotides present in it. E. coli, Escherichia coli, has 4.6 into 106 base pairs. BP means base pairs. And haploid content of human DNA, remember haploid, not diploid. Haploid content of human DNA is 3.3 into 109 base pairs. And what is the structure? Basically, if you recollect from your memory, as a nucleotide, it's made up of three structures or three compounds, chemical compounds. First one is the sugar molecule, deoxyribo. So when we say deoxyribose, like sugar, I said, always ends with OSC. glucose, sucrose, fructose, galactose, lactose, like that. Ribose also is sugar, but in this one oxygen atom is missing, so we say deoxyribose, and it is a pentose molecule. Penta means 5, so 5 ringed structure will be there, so it's a pentose sugar. So first and foremost thing in a DNA or RNA is presence of sugar. So in DNA it is deoxyribose, in RNA it is simply ribose. Oxygen atom is present. That is one. Second is phosphate group. There should be phosphate group linking both the sugar and phosphate. And these two are again interlinked with nitrogen bases. So nitrogen bases, there are two types of nitrogen bases. Both are ringed structure, but one is the double ring purines, another is single ringed pyrimidines. So you got under purines two nitrogen bases, adenine and guanine. We say Ag. Pyrimidines or pyrimidines, cytosine and thymine. Whereas one more is there, that's called uracil. This uracil is present in RNA in place of thymine. So totally 5 nitrogen bases will be there: adenine, guanine, cytosine, thymine and uracil. So a nucleotide has 3 components: a nitrogen base, a pentose sugar, that is ribose in case of RNA, and deoxyribose in DNA, and a phosphate group. So these 3 will be there. Okay, and there are 2 types of nitrogen bases: purines, adenine and guanine. How to remember in the exam? PAG, 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 or GAP, GAP, GAP. Remember, reverse. And pyrimidines, cytosine, thymine and uracil. How to remember? You say UPIC, that is pyrimidine, PY, because they are also purine, so we will put Y. And cytosine, PIC, and T, thymine, so it is PICH. And in place of thymine in RNA there will be uracil, so it's PICU and PICT. So that's how you must remember. Cytosine is a common one. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA in place of thymine. That's what I told you just now. How the picture will be, this is how it will be. So this is a pentose sugar, as you are seeing, and it is attached to phosphate group. This is phosphate group, and the other side, it is the pentose sugar is attached to nitrogen base, like A for adenine, T for thymine, G for guanine, C for cytosine. So this is how the DNA is a polymer. One is attached to each other. And you can also see here 5-phosphate and 3-hydroxyl based on the attachment. That's how we say 5' to 3', we say. So it is a polynucleotide chain. So what is DNA? If someone asks you, you should say polynucleotide chain. A nitrogenous base is linked to pentose sugar with N, that is nitrogen glycosidic linkage, N-glycosidic linkage, to form a nucleoside. So sugar plus nitrogen base. You remember, sugar plus nitrogen base, Sn, is equal to Ns. So nucleoside. Now if you remember this nucleoside, here only it is there, see, nitrogen base and sugar, Ns, nucleoside. When phosphate group is linked at 5th position, the hydroxyl of a nucleoside, through phosphoester linkage, nucleotide is formed. To this we add, what is that? P. We add P, phosphate group, then it becomes NT, nucleotide, or we can say simply NS plus P becomes NT. So keep remembering this difference between nucleoside and nucleotide. I hope I am clear. Nucleoside, we abbreviate it as NS. So N stands for nitrogen base, S for sulfur, sugar, S for sugar, pentose sugar. So nitrogenous base plus sugar is nucleoside. To that, one phosphate group is added, then it becomes a nucleotide. And DNA is polynucleotidal chain, n number of nucleotides. That's DNA. So two nucleotides are linked through 3' 5' phosphodiester linkage to form a nucleotide. That's what I said just now. More nucleotide joins together to form a polynucleotide. In RNA, nucleotide residue has additional OH group present at second position in ribose, and uracil is found in place of thymine. That is important. So differences, DNA and RNA. The sugar is present in DNA as deoxyribose, whereas here the sugar is present in RNA as ribose. So ribose sugar, deoxyribose sugar. DNA contains cytosine and thymine as pyrimidine bases, guanine and adenine as purine bases. And RNA contains cytosine and uracil. This is important. In place of thymine, uracil will be there in pyrimidine base, pyrimidine nitro. Rest all same. DNA is of course double stranded, thanks to Watson and Crick for their discovery. RNA has coiled single strand helix structure, coiled structure. DNA molecules are very large. Their molecular mass may vary from, look at this, so heavy they are, right? RNA molecules are comparatively much smaller, with molecular mass ranging from 20,000 to 40,000 only. So DNA is a heavy fellow. And functional difference, DNA has unique property of replication, DNA replication. Thanks to that, without this property, DNA replication, DNA cannot or would not have replicated, and varieties wouldn't have been formed. Only Xerox copies would have been formed. And RNA usually does not replicate. Of course, usually, but there can be chances. And RNA controls the transmission of hereditary effects. Of course, this is not RNA, DNA. Okay, make a correction. DNA controls the transmission of hereditary effects. RNA controls synthesis of proteins, as I said just now. Now, double helix model of structure of DNA, thanks to Professor Watson and Professor Crick, based on X-ray diffractions data produced by Wilkin and Roslin, proposed this model of DNA. That's why they got Nobel Prize for this. The salient features of this model are: DNA is made of two polynucleotide chains in which backbone is made up of sugar, phosphate, and nitrogenous bases projected inside it. Two chains have anti-parallel polarity. Just now I said one is 5' to 3', another is 3' to 5'. So we say just like railway lines, but they are anti-parallel. Though railway lines are parallel, this we say anti-parallel because of the direction. Okay, because of the direction. 5' to 3' one, another one is 5' to 3', and another one is 3' to 5'. Like as I said, the bases in two strands are paired through hydrogen bonds. This is again blessing for us. Hydrogen bond being weak bond, just little bit of energy if we supply, DNA breaks, and that's how replication, DNA replication starts. So adenine and thymine forms double hydrogen bond. So between A and T, there is a double hydrogen bond. Guanine and cytosine, there is a triple hydrogen bond. Between G and C, there is a triple bond. Two chains are coiled in right-handed fashion, so it's right-handed direction. The pitch of the helix is 3.4 nanometers and roughly 10 base pairs in each turn. Okay, there's a turn like this. When we draw this double helix model, this is the turn, this is the loop. So in this loop, there will be 10 base pairs. The plane of one base pair stacks over the other in double helix to confer stability. So it's just like 1 rupee coins, you keep one above the other, then we say stacks. So like that, the base pairs are in stacks. And you can see the structure. The adenine and thymine has got double bond, guanine and cytosine has got a triple bond. The nitrogen base is attached to sugar, and in turn, the other side, sugar is attached to phosphate group. So this is how the direction, the structure of DNA will be there. And also you can note down here 3 hydroxyl group, 3' and 5' here. and reverse of it. Here it is 3 hydroxyl, here you can see 5. That's why we say it is anti-parallel. And then Francis Crick, of course, thanks to Professor, proposed the central dogma in molecular biology. What is that central dogma? It states that genetic information flows from DNA to RNA, RNA to protein. So this is how the central dogma is there. So DNA undergoes replication and forms RNA. In particular, we say messenger RNA, and that messenger RNA gives a lot of information. So the process of DNA undergoing replication and forming mRNA is called transcription. And mRNA process the secrets, sends the secrets and forms proteins. Then we call this as translation. So roughly you can say DNA undergoes replication and forms mRNA, that is transcription. mRNA converts itself into protein, that is translation. So there are two major steps: transcription, translation. And packing of DNA helix. In prokaryotes, well-defined nucleus is absent, and negatively charged DNA is combined with some positively charged proteins called nucleoids. So in eukaryotes, histones, as you are seeing here in the picture, positively charged protein organized to form 8 molecules. That's why it is octane, right? Called histone octamer. Negatively charged DNA is wrapped around the histone octamer to form nucleosome. So you can see the structure of nucleosome here. Histones are rich in the basic amino acid residues like lysines and arginines. Both the amino acid residues carry positive charges in their side chains. So single nucleosome contain about 200 base pairs, and chromatin is the repeating unit of nucleosome. So you can see here the structure, see DNA, that blue one, histone octamer, that globe-like structure, and histone protein is like a pen, vertical one. So you can see here the structure of nucleosome. So in nucleus, some region of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are called heterochromatin. So euchromatin is transcriptionally active chromatin, whereas heterochromatin is inactive. So we will continue our discussion, how various experiments were conducted by different scientists in search of the genetic material in our next session. Until then, keep writing the